Welcome to the Enzyme Lab, number 3 in AP Bio at Oakton. So what I want to do is give you a quick overview. I know some people missed the lab, some people didn't really understood it because they were not prepared. So in this lab, we are monitoring the activity of one enzyme, and that enzyme is catalase, that we get from potato cells. All cells have catalase, and what we do is we just put the potato with water in a blender, and we basically frappe, break it. The blade breaks all the cells and releases the content of the cytoplasm, and that's where the catalase is. So here is your catalase, basically whatever is in the blender. And this is what everybody gets, and this is our source of enzyme. You could, instead of potatoes, you could do any kind of meat. You could chop out one of your fingers and do the same thing. You have catalase in all your cells. So what catalase does is breaks down hydrogen peroxide that we actually can buy in the store. Right here, hydrogen peroxide. This is going to be the substrate, and catalase is going to break the hydrogen peroxide into simple water and oxygen. Now, to measure a reaction, since we cannot go inside at the molecular level and see this, we need to have a way to do it. So we are going to use a clever device. Here is where you're going to have your catalase and your hydrogen peroxide. Since one of the products is oxygen, oxygen is going to go through, and we are going to collect it in a graduated cylinder that is full of water. So as oxygen bubbles, takes a space here, and we can measure how much oxygen is produced by the reaction over time. So in activity A, what everybody has to do is what we call the baseline, so we want everybody to see it. So you put a certain amount of catalase, a certain amount of hydrogen peroxide. In our case, we use 10 mL of catalase for all the reactions, 10 mL of catalase, and 5 mL of hydrogen peroxide. This is going to be pretty much the standard for all the lab, for most of it. And we are going to test the effects of temperature, pH, and enzyme concentration on the progress of this reaction. So in the first activity is basically you mix the catalase, the hydrogen peroxide, and over a period of time you see what's going to happen. What's going to happen is that everybody saw this, and I posted the data so you can see it on Blackboard. Over the first minute, between time 0, the moment you mix it, time 0 is when you see the first bubble come out. In the first minute of the reaction is where most of the oxygen is released. In the first minute is when most things happen. So if you graph this, and that's the first graph that you have to do, we are going to call this graph a progression curve, because it's going to show us the progression of the lab, and this is what we do in part A. Here we are going to have time, 1 minute, 2, 3, 4, 5, 6, 7, 8, 9. Most reactions ended up around 10 times 0. And here is the mLs of oxygen produced in milliliters, 10, 20, 30, 40, 50. And what most people are noticing that in the first minute you have like about 20 mLs, then starts second minute, third minute. Then notice in the first minute is when you put the most, and then by minute 5 you are close to 40, and then very slowly the oxygen keeps on increasing, but very, very slowly. And then you get to a point where you get, if we go to 12, 13 minutes, you get exactly the same reading. Let's say 49, 49, 49, 49. we usually stopped it when you got two readings the same, because that means that there is no more oxygen being produced, or is being produced very, very slowly. So this is what the progression curve is going to look like, and it shows you the main part is that at the very beginning is when the reaction is happening the fastest, when the most oxygen is being produced. So in this case, you can say that in the first minute, 20 mLs of oxygen were produced. That's a very fast reaction rate. So this is the baseline that you have. Now, the main thing to do in this part, you have to be very comfortable calculating reaction rates at different time intervals to see how the reaction progresses. So you have to do that in your lab. Please follow the directions. I explain everything very carefully, but just in case, reaction rate. For example, if we want to calculate the reaction rate at minute 1 here, and we're going to keep the numbers simple, we're going to assume that it's 20 mLs at 1 minute. So reaction rate, or the rate of reaction, is always delta Y, delta X, the change in Y divided by the change in X for that interval. So here the interval is between 0 and 1. Change at 1 is 20 minus 0, of course, divided by the change here in time, so it's 1 minus 0. Basically 20 mLs of oxygen produced per minute. Remember, this is the same thing that in math you do that you call the slope, or rise over run. And just to remember always, every calculation of rate or slope that we're going to do through the year is the same, rise over run. And remember that first you have to rise out of bed before you can start running. So that's a nice mnemonic. If I ask you, for example, to calculate the reaction rate between minute 2 and minute 4, what do you have? Here is minute 2, here is minute 4. So rate between minutes 2 and 4 is going to be first the change here, minute 2 is 30 mLs, minute 4 right here is 40 mLs, so 40 minus 30 divided by the change in X, 4 minus 2. So this gives us 10 divided by 2 equals 5 mLs of oxygen per minute. So as you can see, if we calculate the rate of reaction for the first minute, for the interval 0 to 1 here, the rate of reaction is 20 milliliters of oxygen produced per minute. Think about it, 20 mLs of oxygen produced per minute. Between minutes 2 and 4, the amount of oxygen produced, the rate of the reaction, is only 5 mLs of oxygen per minute. So this is why we have to calculate rate of reaction at different intervals to get an idea of how the reaction is progressing. A point of interest here. Let's calculate the rate of reaction, for example, between minutes 9 and 10. Minute 9, minute 10. If you calculate that rate for 9 to 10 minutes, delta Y, the change here, since it's 49 and 49 mLs, 49 minus 49, and the change in time, 9 to 10, that is technically 0. 0 rate of reaction. Obviously, no more oxygen produced, that means a reaction is not happening. So the rate of reaction here is 0 mLs of oxygen per minute. So this is very interesting. So if you look at any of the data sets, the fastest rate of reaction is always in the very first minute. And why is that? If you look at the reaction, during the very first minute, you have all the enzyme is always going to be there, and you also have all the substrate. All the substrate is available, and you have your beautiful catalase ready to act. So during the first minute, plenty of substrate. As this encounter and the reaction progresses, what's happening after the first minute? The amount of hydrogen peroxide decreases, the substrate decreases, and the enzyme keeps on going around, bouncing, trying to find more hydrogen peroxide. And that's why the reaction slows down, because they have to find each other. And the only way that reactions happen is when they crash into each other with collisions. So that's basic baseline, so you understand what's happening in a reaction. So that's what happens with A. You have a lot of questions to answer just to make sure that you understand all this process. So calculate reaction rates and get good at it and understand, try to visualize the process. The second part of the part B of the lab focuses on, I'll just redo the whole thing. Part B of the lab focuses on the effect of temperature on the reaction, and we use 3 temperatures: 4 degrees Celsius, that is cold, 22 degrees Celsius, that is the room temperature, and then catalase that had been in a water bath at 50 degrees Celsius. 50 Celsius is a very, 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 very hot shower. Those showers that you get in and you need to get out. A little bit hotter than a hot shower, so it's high up there. What we saw during that activity is that the curve for 22 degrees is pretty much like the baseline, nice, optimal conditions. At 4 degrees, and we are using exactly the same amount of catalase and hydrogen peroxide, but at 4 degrees, the reaction when, and I'm going to do it in another color here, at 4 degrees, the reaction when something like this. Notice what happened during the first minute right here. 22 degrees, you produce a lot of oxygen. The curve at 4 degrees, the amount of oxygen produced during the first minute, much, much lower. So that tells you right there, the rate of reaction during the first minute is much, much lower. You can see this in the graph that you're going to draw, but you can also just see it directly in the data table during the first minute. It tells you immediately. The other line that we had to do is what happened at 50 degrees. And at 50 degrees, there was absolutely no reaction whatsoever. So you never had any oxygen being produced. So this is kind of the type of curve that you are, for most of the part, you're going to get for temperature. I have a couple of important considerations here. For the optimum conditions, at some point the curve becomes flat, no more oxygen being produced, and there is no more oxygen being produced because if you don't have any more substrate, there's nothing to break. So that's why the reaction ends there after a given time. Now notice that the data that we have, 50 degrees, obviously here temperature did some damage and the enzyme has been denatured. But at 4 degrees, the reaction rate is much lower. Everything is happening much lower. And the reason it's happening much lower, again, if you read your lab, you'll get it, is because all the molecules have kinetic energy. The reactions depend on collisions, and at 4 degrees, everything is happening very slowly. So catalase and hydrogen peroxide, even though we are shaking them a little bit in the reaction chamber that we have, you are constantly twirling around, moving it, still not enough, and reaction rates are slow because the kinetic energy of the molecules is low. As you increase temperature up to 22, molecules start moving faster, they encounter beautifully, perfect. What happens if you keep on increasing the temperature beyond the optimum for that particular enzyme? Remember that all enzymes are proteins that have a particular shape and that the active site is a very definite place in the molecule. As you go up, temperature increases, and if you recall, we have primary, secondary, tertiary structure of proteins. Primary structure is just the sequence of amino acids. Secondary structure is the beta pleats or alpha helices that are maintained by H bonds. And the tertiary structure is the full three-dimensional shape of the molecule that is maintained, among other things, by ionic, disulfide, and hydrophobic and hydrophilic interactions. Now, as you increase temperature, think of water. What happens to hydrogen bonds in water as you heat up the water? The hydrogen bonds start, the molecules move faster, and the hydrogen bonds break with temperature. As temperature increases, hydrogen bonds break, and the hydrogen bonds that were helping maintain this structure disappear, and when you have no hydrogen bonds, the molecule just goes flat. So if I have my nice beta pleats here in my molecule, you have nice beta pleats there, you can see again the hydrogen bonds between two sides here holding this molecule in this shape. If I break those hydrogen bonds there using heat beyond what is acceptable, the molecule flattens and it loses its shape. So please remember, temperature destroys hydrogen bonds, and when you destroy the hydrogen bonds, the shape of the molecule is gonna change, and at some point it's gonna totally denature. Now, until it denatures, other things might happen and might start losing its shape, so activity might decrease. So you need to think of all these possibilities. So this is the progression curve for temperature. Now, what are you gonna do once you have this progression curve? Once you have this progression curve in your lab, I'm gonna erase part of it to have room here. Once you have your data for your progression curve, you are gonna draw what is called a rate curve, where you are gonna put basically how the rate of reaction changes under different temperatures. So here you're gonna have temperature. Here you're gonna have rate of reaction. And the rate of reaction, we're always gonna calculate it for the very first minute. How much oxygen was produced during the very first minute at this temperature, at this temperature, at this temperature. So when you do this type of curve, temperature in Celsius, rate of reaction in mLs of oxygen per minute, please remember your units. What you're gonna have is temperature 0, 10, 20, 30, 40, 50. And rate of reaction, you're gonna go 10, 20, 30, 40, 50 about. You have a nice table that I guide you, so read every word and follow the sequence of events in the lab. I have explained every step carefully in the lab itself. Once you calculate, that is very easy, the rate of reaction during the first minute for these three conditions. So for example, for 50 degrees, the rate of reaction is gonna be 0. because there was no oxygen produced. 50, right there, 0. For 22 degrees, the rate of reaction was something like, for 22 degrees, the rate of reaction in the first minute was something like 20 mLs of oxygen, or something in between 20 and 30. And for 4 degrees here, the rate of reaction was much lower. Here is 4. Let's put the dot right there. So when you make your curve, what are you going to see? This is how the effect of temperature on the rate of reaction. And this is the usual curve that you see in your textbooks or anywhere where you are seeing the effect of temperature on enzyme activity. This is called the rate of reaction curve. Rate curve or rate of reaction curve. So this is how you construe it. We have only 3 data points because we only have time in the lab to do 3 things. If we have more time in the labs, and you'll do this in college when you have 3-hour labs, you can have curves all in between for different temperatures, and you can construe or build a more accurate reaction rate for temperature. That's what you do. So this is what you're going to do for part B, that is temperature, part C, that is pH, and part D, that is the effect of enzyme concentration on the rate of reaction. Now, before I go over a little bit what's happening in the other ones, you have 3 curves here. You have 22 degrees, you have 4 degrees, and you have 50 degrees Celsius for the reaction. Here is the progression because it's time, and here is the amount of mLs of oxygen produced. What is the dependent variable here? Remember, D for data. Dependent is the new data that you are generating. And the new data is always here. What is the independent variable? The one that you are changing. And what is the thing that you are purposely changing here? You are changing temperature. So please, time is not the independent variable here. This is a progression curve. It has to happen in time. Please don't get messed up like in physics. That is always time here, and that's your independent variable. Here we're measuring the progression of 3 different conditions, 3 different temperatures. Temperature is your independent variable. That allows us then to build this other curve. So temperature. Next was the effect of pH on the reaction. And if you graph, I'm just going to make a raw graph here. For the effect of pH, we have exactly the same situation. We have time increasing. We have oxygen produced in mLs. And for pH 7, which is the normal, just mixing the catalase and hydrogen peroxide, we get this kind of curve, pH 7. When we add a buffer to this, we mix the catalase, hydrogen peroxide, and then a buffer. I had 2 buffers, pH 3, not 4 like it says in your lab, pH 3 and pH 11. So we use pH 3 and pH 11. What everybody found out for the most part is that pH 3, no arrow, we don't have data going there, pH 3 did not get a reaction for the most part, or it got 1 ml of oxygen in the whole time. pH 3, and then for pH 11, we got something like this, pH 11. So same principle as before, optimal conditions, 11, the enzyme is not working very well. That means it's starting to change shape under these conditions, and the active site is not quite perfect. pH 3 definitely changed the shape of the molecule altogether. With this data, again, you're going to calculate for minute 1 here the rate of reaction, and you're going to plot the rate of reaction again for those 3 pH in a rate graph with pH here and rate of reaction in the first minute over here. So you're going to get something like that. Please make sure that you are scaling properly. These are not bar graph, these are continuous measurements. If we had time, we could always measure every single pH, how it affects the enzymes. So they are all nicely scaled. I want to stop right now for a second to point something that is going on in all these graphs that is important. For the most part, the one under the optimum conditions, either we got to a flat line because all the hydrogen peroxide was consumed, or we had to stop the reaction because we were running out of time in class and we couldn't keep on waiting until everything was consumed. So sometimes some groups really got to 2 measurements that were exactly the same. Sometimes I stopped it when it was like increasing by 1 ml non-stop, 33, 34, 35. Now, for those people that got the flat line at the end, so that the data was totally, all the hydrogen peroxide was consumed, if you notice, and this is true for pH, for temperature, and for enzyme concentration, the curves, under the less optimum conditions, the progression curves, you are producing much less oxygen. If we had the time to let this reaction go for 24 hours or so, and we were able to stay here, what would happen to curves like pH 11? What would happen to curves like the temperature at 4 degrees? If you go back to the equation, if you have a particular amount of hydrogen peroxide that you start with, in this case, everybody measured exactly, I hope, 5 mLs. If you measure exactly 5 mLs, everybody has the same amount of substrate, and because this is a regular reaction that is balanced and substrate products, nothing disappears, conservation of matter, because you have a particular amount of hydrogen peroxide, everybody is supposed to end up producing exactly the same amount of oxygen. Once all the hydrogen peroxide breaks, same amount of oxygen. So, what's going to happen here? Right here, if this is close to the 50 mLs of oxygen that most people were getting, here we are not even close to 50, and the reaction is going very slow. So, if we allow this enough time, what's going to happen? Is keep on going, very slow increasing, very slow increasing, very slow increasing, until, I'm going to just jump, it reaches the 50 mLs here. At the end, every single one of these reactions, if we would have allowed them enough time, they would have reached the same amount of oxygen produced, because we started all with 5 mLs of hydrogen peroxide. So that's an important concept. You start with the same product, you have to have the same, backwards. You start with the same substrate, hydrogen peroxide, you should get the same amount of oxygen. If you let the reaction go long enough so all the hydrogen peroxide is destroyed. Good? Now, food for thought. What happens if instead of 5 mLs of hydrogen peroxide, you do it with 10 mLs of hydrogen peroxide? What would happen to the amount of oxygen produced? It would double. Twice as much, twice as much oxygen. What happens if instead of 5 mLs, I use 2.5 mLs of hydrogen peroxide? What happens to the amount of oxygen? Also, it's going to decrease. So those are things that you need to play with your mind and be able to, on the fly, kind of realize when we are doing enzymes or talking about enzymes. What happens when you increase temperature? What happens when you change pH? What happens when you have more substrate? What happens when any of these things happen? So this is the baseline for the rest of the year. The last activity that we had was calculate enzyme concentration. And for enzyme concentration, we did same thing, 5 mLs of hydrogen peroxide, and we tried 3 different amounts of catalase. We couldn't measure concentration, but we measured the amount of catalase that we use. So again, we have time in minutes, and oxygen produced in milliliters. And if I can flatten it, this is the curve for 15 mLs. mLs and 5 mLs. And again, the idea is that if you allowed enough time, all these curves, of course, are going to converge at the maximum amount of oxygen produced. So again, you decrease the amount of enzyme, you have less hands to do the work. Think of my analogy of the making peanut butter and jelly sandwiches. Fewer hands, you're going to go slower. If you get all the class here making PB&Js, we are going to produce a lot of sandwiches much faster. So this is what you need to master and you need to understand. So we hit all the major things. I explained the effect of temperature. I want to make sure we understand the effect of pH on the enzymes. It's in your lab again, but a visual never hurts. As you know, proteins have the primary, secondary, tertiary structure. And for secondary, you have mostly H bonds or H bonds being responsible. For the tertiary structure, H bonds help, but also you have disulfide, and more importantly, you have ionic bonds. That's between a charged amino acid, a positively charged amino acid, and a negatively charged amino acid. And when we are talking about this, we are always remember talking about the R groups of those amino acids. So what happens if you have a change in pH? So let's make a funny shape here. And let's say that this shape here is being maintained because here is a positively charged amino acid and here is a negatively charged. So here we have a nice, let me make it in a different color. You have a nice ionic bond, an ionic bond kind of attracting the positive and negative. So what happens when you change the pH? Let's say, think of pH 3. pH 3, that means 10 to the minus 3 molar concentration of hydrogen ions versus 10 to the minus 7 concentration at pH 7. So obviously, at pH 3, you are going to have more hydrogen ions present in the solution. You are changing that. Now, when a protein has a particular shape, all these shapes are functional at usually the pH that is supposed to be. Normally between 6 and 8, they hold their shapes pretty good because that's the normal conditions in the cell. Their interactions are balanced by the amount of hydrogen ions at that pH. Now, if I do pH 3, I'm going to add more hydrogen ions. That means more positive charges. So I have this situation here, and I'm going to increase the amount of positive charges all over because I'm changing the pH like from 7 to 3. That's like 10,000 more hydrogen ions. So what's going to happen when you have this? this was a negative, this was a positive, they were highly attracted, and they were bonding. But notice now that we have all these positives surrounding this negative. There is competition for this. This guy not only has this positive all the way over there to bond, but you have all these other positive charges surrounding it. So what is always the easiest way? Hey, you are next to me, let's bond. So the bonding happens here with any of the charges around, and this bond technically breaks. And if this bond breaks, you can visualize that this V shape here is not going to hold, and the molecule is going to go flat. This is how pH changes things, interferes with the bonding between ionic bonding and also hydrogen bonding. Because remember, hydrogen bonds is between the slightly positive and the slightly negatively charged R group of another one. So anytime that you mess around with the charges, it's going to change how these bonds are formed, and it's going to change the shape of the molecule. Remember, hydrogen ions, when you get more acidic, you get more positive charges. When you get more base, you have fewer positive charges, and you have more hydroxyls, which are negatively charged. Regardless of which pH you are going away from, you are having an imbalance of charges. So I call these guys usually relationship breakers. You are in a nice, stable relationship with somebody, and here comes a bunch of jerks and surround either one of the couples, interferes, and that couple breaks up. That's pretty much what happens here. So that summarizes the whole thing. Please think about it. I'm done with this. At the end of the lab, there is a nice, beautiful free response problem. Please take it seriously. Do your work. Try to do it. And if you have any questions, I'll be here as always, Cougar time, and after school Monday and Wednesday for sure. Every other day is just potluck. I don't know if I'll be here. All right, so I hope this helps. I'll see you next week.